Welcome everyone to the 2020 Safadi Lecture, hosted by the Safadi Program of Excellence in Clinical and Translational Neuroscience at the University of Chicago. My name is Issam Awad. I am the John Harper Seeley Professor of Neurosurgery and Neurology, and I am honored to direct the Safadi program. This program was established through a generous gift by His Excellence Muhammad Safadi in gratitude institution and in recognition for the intimate collaborations among our clinicians and scientists. The program aims to facilitate multidisciplinary interface and create opportunities for collaboration among scientists and clinicians engaged in the diagnosis and treatment of neurological disorders. It catalyzes partnerships with groups across the university, enabling cross-disciplinary thinking that allows us to tackle the complex problems in the field. I wish now to recognize the eminent professors who serve as the advisory committee to the Safadi program. David Frim of Neurosurgery, Sham Prabhakaran of Neurology, John Monsell of the Grossman Institute for Neuroscience, Mark Siegler of the Buxbaum Institute of Clinical Excellence, and Julian Solway, Director of the Institute of Translational Medicine. And I want to thank them really for the advice they give us and the direction in the priorities of this program. I now present Dean Kenneth Polonsky, the Richard T. Crane Distinguished Service Professor, Dean of the Division of Biological Sciences and the Pritzker School of Medicine, and Executive Vice President for Medical Affairs at the University of Chicago, who will deliver the institution's welcoming remarks and introduce Mr. Safadi.
Great. Well, thank you very much. Can everybody hear me properly?
Yes.
Good. Okay, great. So welcome everybody. And obviously events like this are special at any time in for an institution and for our academic programs to be able to have the opportunity to hear distinguished experts like Dr. Iadecola speak on important topics. But I think under the current circumstances where we're not able to get together in person, events like this assume an even greater importance. So we thank you very much, Issam, for your leadership in this program. And we thank Dr. Iadecola for being with us today and so that we can benefit from your knowledge and experience. It gives me particular pleasure to acknowledge Mr. Safadi, who is a real supporter and friend of the University of Chicago and particularly of our programs in neuroscience and neurosurgery. He's a very distinguished person, a successful businessman, philanthropist, and public servant. He began his career in the private sector, was a very successful business person in property development and business aviation. And then he was elected to the Lebanese parliament in the year 2000, and he served in that body as until 2018. He's also served his country as Minister of Public Works and Transport, Economy and Trade, and most recently as the Minister of Finance through 2014. Mr. Safadi supports the development of Lebanese civil society and the reform of state institutions through the Safadi Foundation. He also serves on the Dean's Council of the John F. Kennedy School of Government at Harvard University and on the boards of several American educational institutions in Lebanon. He's also a member of the Middle East Advisory Board at the Woodrow Wilson International Center for Scholars, and he was granted the Order of the Knight from the French government, the Award of the Cross of the Order of Merit of the Federal Republic of Germany, and the Order of Civil Merit from the Kingdom of Spain. So he's an extraordinary, distinguished person, and we're very delighted to be able to count him as a friend and a colleague of the University of Chicago. So thank you very much, Mr. Safadi, for being here, and he's now going to present a few remarks. Mr. Safadi.
Thank you. It's me to speak now?
Yes.
Yes.
Okay. Ladies and gentlemen, it's such a privilege given by God to humans who are in the medical profession and medical research to save lives and improve its quality. I'm privileged and honored by the University of Chicago Medicine and Biological Sciences, represented by Dr. Aysam Awad and his research team, to contribute and support such a noble cause. So many people dedicate themselves trying to save lives, whereas so many lives by intent or by negligence. The Beirut blast on 4th of August 2020 has killed hundreds and injured thousands of innocent people, destroying homes and dreams. Even hospitals were not spared. Some they say is negligence. Others they say is intentional. We don't know what's which, but what happened has been vicious, cruel, and unnecessary. Let's hope that saving lives will overcome the destruction of lives and leave it to nature to do its course. I would like to thank you, our lecturer, Dr. Costantino Iadecola, the University of Chicago Medical School, Dr. Aysam Awad and his research team, and all those who made our meeting possible today. Thank you all for attending this lecture by Zoom. I'm sure we will all benefit from the valuable information and knowledge that will be shared today. Thank you very much.
Thank you, Mr. Safadi. Thank you, Mr. Safadi. And we appreciate your kind words and your humanistic commitments. We remain grateful for your friendship and support of the noble aims of this program of excellence carrying your name. It is befitting, with your permission, to take a moment of silence and respect for the many victims of the recent tragic Beirut explosion. Thank you. Now I am pleased to provide a report on the 2020 Safadi Pilot Award. 13 applications for pilot awards were received this past year. These were reviewed by 3 eminent reviewers, 2 within the institution and 1 from outside. They graded each submission in 6 domains. This year's submissions were particularly competitive, scoring an amazing mean of 2.7 on a scale of 1 to 5. The winning award scored 2.0 with 3 proposals tying for second. Congratulations on a truly exceptional winning proposal, bridging developmental neurobiology, endocrinology, diabetes, genetics, neurophysiology, and the use of innovative patient-derived cerebral organoid model. The winning proposal embodies all the goals and values of the Safadi program and is entitled Examining the Neurodevelopmental Role of KC and J11 in Patient-Derived Cerebral Organoids. I am privileged to present remotely this very nice award to Siri Atma Greeley, MD, PhD, Associate Professor of Pediatrics, who received the award, and Dr. Greeley will give us an update on the progress of the project.
Thank you very much, Aysam, and I am very honored to have been awarded this the funding that, as Dr. Awad mentioned, I think really does embody the goals of the Safadi program in at least two major ways. One is the multidisciplinary collaborative nature of the work, and then also the fact that it's not very far at all from the patients who really are dealing with this very difficult disease, and we're really trying to help understand that disease better to help them in their lives. I am a pediatric endocrinologist, and I've kind of become a geneticist by working with Graham Bell, Lou Phillipson, and others on our monogenic diabetes program, which actually Dr. Polanski spent many, many years developing a lot of what we know about these rare forms of genetic types of diabetes. My particular expertise is in what we call neonatal diabetes. So as you might imagine, when a baby develops diabetes, that's a very unusual situation where it turns out to, in almost all cases, be due to specific mutations in specific genes. And the most common of these is related to our project, which is mutations in the ATP-sensitive potassium channel from two different genes, where the most common is this KCNJ11 gene. And so we have about 300 patients with these mutations from all over the country and beyond. And the really exciting thing about this work has been that by discovering the genetic etiology of their disease around 2006, it was quickly apparent that these patients, instead of being treated with insulin, which is all that anyone knew before, they could now be treated with sulfonylurea pills that was much easier and controlled their diabetes very well. However, the problem is these channels are also expressed widely in the brain, where they cause a spectrum of neurodevelopmental disability that in some cases is very mild, but unfortunately in many cases can be quite severe. And so even though we, through the genetics, had helped to essentially cure their diabetes, these neurodevelopmental problems that they have become really a major concern and difficulty for these families. And so this project, which by the way, I'm representing Gokhan Daljing, our postdoc, Alfredo Garcia, and Andrew Treba, who really helped to do most of the work. And what we were able to do is to confirm that when you take patient samples, so we can just take blood from these patients, and by using stem cell technology, develop induced pluripotent stem cells where we condense their blood cells to become brain cells, essentially, that in a dish start to organize in a collection that we term cerebral organoids. So it's not really the full complexity of the brain, but gives you a sense of how things are developing and working. And what the work, thanks to the funding of the Safadi program, has been showing is that we've confirmed that these patients from very early in this brain organoid development start to have defects in how that happens, and they're not able to develop a stable neural network in comparison to controls. And this corresponds around the time of what we would expect to be the generation of the neural precursors. But what has been very interesting is that these same sulfonylurea drugs that we use to help the channels to work right for the diabetes, when we add that to these cultures very early on, that seems to rescue at least part of this phenotype and allow the cells to develop this neural network in a more normal way when they have the drug present as to when they don't. So this is really important foundational work to help these patients so we can try to understand how we may even eventually be able to prevent some of this neurodevelopmental disability in the future, we hope.
Thank you for this very nice report. And on behalf of the Safadi program, we are very glad that we were able to help you get this work to another level.
Thank you very much.
We will get the plaque to you. We apologize about the lack of personal delivery. The annual Safadi Lecture celebrates neuroscience excellence at the University of Chicago, addressing topics of great relevance by preeminent experts in the field. I am now delighted and honored to introduce our 2020 Safadi Lecturer. How we wish to have been able to receive him in person in Chicago, but we truly appreciate his willingness to present his lecture remotely, seizing an opportunity to welcome a greater number of attendees in real time with us today from around the world, and through recording and posting the lecture on the Safadi program and the Safadi Foundation web pages for future viewing. Costantino Iadecola is a clinician scientist who is an internationally recognized expert in the field of cerebrovascular diseases and dementia. He received his MD degree from the University of Rome, Italy in 1977. He then completed neurology residencies both in Rome and in New York, and joined the Department of Neurology of the University of Minnesota, where he rose through the academic ranks, serving as professor and vice chair for research. In 2001, Dr. Iadecola was recruited back to Cornell as a distinguished professor of neurology and neuroscience and director of the Division of Neurobiology. Since 2012, he has held the Parrish Titzell Professorship of Neurology, and he serves as director and chair of the Feil Family Brain and Mind Research Institute, a new department at Cornell focused on basic and translational neuroscience research. Professor Iadecola is a pioneer in establishing the concept of the neurovascular unit. He has elucidated the role of neurovascular dysfunction in neurodegenerative diseases and the role of innate immunity and the microbiome in ischemic brain injury. He has published over 500 papers and chapters and is listed as one of the world's highly cited researchers for ranking in the top 1% among authors in the field of neuroscience and behavioral sciences. He has a truly impressive H-index of 114 among the highest scholars in any field. A member of the Association of American Physicians, Professor Iadecola has received numerous awards, most recently being named Distinguished Scientist by the American Heart Association. He has twice received the prestigious Javits Award from the National Institute of Neurological Disorders and Stroke. Costantino, there is also a very nice plaque for you that will find its way to New York along with our warmest appreciation. And it is now my honor to welcome you to present our lecture today.
Thank you. I'm going to start sharing. Let me first thank you all for this invitation and for this to participate in this distinguished program. I was not aware of His Excellency Safadi in this promoting this translational neuroscience, which I think is the way of the future, because we do a lot of work in the laboratories, and we need to bring this work back to the clinic to make an impact on what's going to be one of the major diseases of the future, dementia and so on. It's already hitting us hard, but it's going to be even worse as the population ages. So I'm going to share my screen now.
While Dr. Iadecola is doing this, I would like to ask the audience if they have any questions to please address them to me. You have the option on the Q&A. Do not send them to all attendees, just send them to my attention, and there will be an opportunity to share a few questions at the end, which I will proceed with. Thank you, Cost.
Okay, am I projecting correctly?
We are seeing the slideshow thing. You need to put it on the regular, yeah, swap displays. That's perfect.
Okay, great. So I'm gonna, this is the title of my talk. I'm gonna talk about the role of vascular factors in cognitive impairment, and particularly focusing on Alzheimer's disease and other causes of dementia. Now, to briefly outline my talk, I'm going to tell you a little bit about the concept of dementia through the ages. And then I'm going to give you a little bit of information on how the blood vessels of the brain are organized, and they seem to be working. And then I will focus on the impact of this vascular dysfunction on cognition. And we're going to present examples from three major conditions: hypertension, Alzheimer's disease, and excessive intake of dietary salt, how that affects cognition. And finally, we're going to kind of have a correlate among these different conditions, and establish the concept that some of this cognitive impairment does not necessarily derive from blood flow to the brain dependent factors, but there are also other aspects that may contribute to the cognitive impairment. So let's go to dementia. Most of my talk is going to be based on experiments derived from animal studies, but in agreement with the translational nature of this program, I will make patient-related correlates every chance I have, just to kind of verify that whatever we see in animals may also be occurring in humans, which is actually the scope of the whole program. So let me tell you a little bit about the history of dementia. So at the time of Alzheimer's, in the early 1900s, the major cause of dementia was thought to be hardening of the arteries, which somehow led to cognitive impairment. However, in the 80s and 90s, with the unveiling of the molecular pathology of Alzheimer's disease, for example, the amyloid precursor protein, the genetic mutation of the amyloid precursor protein leading to familial form of Alzheimer's disease, this molecular revolution in the field of dementia led to a polarization of the field towards Alzheimer's and neurodegeneration. And so nobody really cared much about vascular factors in dementia anymore. However, over the past 20 years or so, there has been an increased appreciation for the role of vascular factors in dementia. And one of the major lines of evidence comes from comparing clinical diagnosis of dementia in the doctor's office with the pathological diagnosis after the patient dies and then an autopsy is performed. Now, in the case of the clinical diagnosis of dementia in the doctor's office, the majority of cases are thought to have Alzheimer's disease based on clinical criteria. However, when the pathology is examined, this group of patients got a combination of a wide variety of pathology, the majority of which is a combination between vascular factors, vascular alteration, and Alzheimer-related alteration, which means plaques, amyloid plaques, and neurofibrillary tangles. And now this 53% number get higher and higher as the population gets older. So in the 75 to 80-year-old can go as high as 60–70%. So blood vessels, have a profound impact on the expression of the dementia in combination with Alzheimer pathology. Now, a curious event has occurred really very recently, so I call this Back to the Future, because a recent study in which there was a proteomic-wide study in one of the largest cohort of demented patients, actually in Chicago, at Rush, found that intracranial atherosclerosis is a major contributor to Alzheimer and the cognitive impairment through mechanisms which are very similar to the one caused by tau and other factors. So we're going back to Alzheimer's point that hardening of the arteries, is really very, very important in the mechanism of cognitive impairment, justifying, the emphasis on vascular factors in dementia. Now, this is not surprising that the vessels are very important in cognition because, the brain does not have any fuel reserves and relies on the second or millisecond by millisecond delivery of blood flow in order to function. So it's like having, a race car without a tank. So a race car uses a lot of, fuel, but does not have a way to store it. So it needs to be refueled on a millisecond by millisecond basis. And so there is a lot of emphasis in the brain regulation for making sure that there is always blood flow getting into the brain. One of the mechanisms that assures that this occurs is called functional hyperemia, which is the ability of neurons to tell the blood vessels that they need more blood, all right? So neurons generate vasoactive signals that travel to the blood vessels, produce vascular relaxation, and increase blood flow only to the regions where there is increased activity. And this correspondence between neural activity and blood flow is so temporal and spatial specific that is used to localize brain function. So what we now call functional brain imaging is nothing else than a proxy of the blood flow changes reflecting neural activity. So neural activity is a major controller of blood flow to the brain. The other major regulation factor is autoregulation, which is the ability of the brain to maintain a more or less constant blood flow despite changes in arterial pressure. Now, why do we need autoregulation? Well, we need it because blood pressure normally, in a normal individual, changes by 40, 50 millimeters of mercury depending on what we're doing. And since blood pressure is the major driver of blood flow to the brain, if there was no autoregulation, there would be a changing baseline in the brain. And so every time the blood pressure would change, also the blood flow to the brain would change. So autoregulation provides some kind of stability to the delivery of blood flow to the brain, so that these other mechanisms like functional hyperemia can work effectively because they have a stable baseline. Then there is another very important component in this regulation, which is provided by the endothelial cells that line cerebral blood vessels. So endothelial cells are very important mechanical and chemical transducers of signals from the blood and from the brain, resulting in adjustment of vascular resistance. So they can regulate blood flow by relaxing the vessels or constricting the vessel. And one of the major factors regulating this constriction and relaxation is nitric oxide, which is produced by the endothelial nitric oxide synthase enzyme, and is pretty much regulated by the muscarinic receptors activated by acetylcholine. So I'm telling you this now because this is going to come up later in the talk. So I want you to kind of pay some kind of attention to the endothelial cells and the production of nitric oxide. So these factors work together to make sure that the brain always gets the blood flow that it needs. However, the vessels of the brain do more than regulate blood flow. They also assure the blood-brain barrier exchange of substances. They also provide trophic supports to neurons and glia, pretty much like neural and glia provides trophic support to the vessels. So there is a symbiotic relationship between the vessels and the brain cells. Furthermore, the vessels serve as a conduit for immune cells that continually traffic in and out of the brain, surveying the cerebral microenvironment, and are responsible for some of the immune regulatory events that occur in the brain. And finally, as recently kind of emphasized, the vessels of the brain are critical for waste removal. The brain is very metabolically active and makes a lot of waste. And so this waste needs to be removed. Now, one of these wastes is consisting of proteins that the brain makes, and they are very bad for it. For example, A-beta. And so A-beta is produced during synaptic activity, needs to be cleared. And one of the major mechanisms of clearance of A-beta is through the blood vessels. So it's not surprising that if factors that affect the blood vascular functions, like for example, vascular risk factors like dietary salt, hypertension, and even Alzheimer's pathology are able to alter these regulatory events, leading to brain dysfunction and damage. So now I'm going to provide you some mechanistic insights in how these different factors are able to alter cognition and through this vascular mechanism. Now, hypertension is one of the worst things that could happen to the brain. Hypertension promotes atherosclerosis that leads to stenosis of the kind of occlusion and stenosis of the arteries in the neck, thereby reducing globally blood flow, which may also lead to cognitive impairment. Hypertension leads to microbleeds, thousands of microscopic hemorrhages provided by seepage of the blood outside the blood vessels, and also microinfarct, which are equally small lesions that cannot be seen with the naked eye, but they can be seen under the microscope. However, the classic change that hypertension induces in the vessels of the brain is called lipohyalinosis, which is a process of kind of destruction and alteration of the vascular wall in arterioles located in a very peculiar region of the brain, which I call like a no man's land, right? Is an area in between two different arterial territory, which doesn't quite get the blood flow that it needs at all times. Furthermore, these vessels providing the blood flow are terminal arterioles, so there is no way that there is collateral circulation. So the blood cannot come from any other way than through these vessels. So when you have things like this happening here, you get this kind of situation. You have white matter disease. Essentially, you have a degeneration, demyelination of the white matter tracts connecting the different areas of the brain. So de facto, you have a disconnection syndrome. So the right hand doesn't know what the left hand is doing. And so the cognitive areas are disconnected, and there is the classical executive dysfunction and psychomotor slowing that we observe typically in patients who have this kind of vascular type of cognitive impairment. Now, hypertension also alters the regulation of blood flow. All those nice things that I told you before, they make sure that the brain always gets enough blood, that they're altered in hypertension. So there is a problem with autoregulation that gets kind of shifted and no longer works properly. There is opening of the blood-brain barrier. There is also endothelial dysfunction, so the endothelial cells are unable to provide the blood flow through those mechanisms. And also this alteration in increasing blood flow produce neural activity. So as you can see, all facets of vascular regulation in the brain are profoundly disrupted by hypertension. So the question is, how does that happen? So a number of years ago, we began to explore the mechanism whereby hypertension could affect, and also other aspects. So we had to set up methods to look at the regulation of blood flow to the brain. So to do that, we do this in anesthetized mice, in which a window, an opening is drilled over the somatosensory cortex, and blood flow is monitored in this area of the brain which is being exposed. Now, to activate the brain, to look at the neurovascular coupling, which is the increase in blood flow produced by neural activity, we use the whisker barrel cortex model. So in rodents, whiskers are very important. It's like a very important sensory organ. So mechanical stimulation of the whiskers will lead to activation of restricted group of neurons in the region of the somatosensory cortex called the whisker barrel cortex. So if you monitor blood flow over this region of the brain, while you're stimulating mechanically the whiskers, you're going to see this increasing blood flow highly restricted to the areas that is activated, as shown by field potentials and so on. If you monitor the blood flow changes over time, these are locked to the stimulus, and they are independent of any change in blood pressure, blood gases that may be affecting the blood flow change. Furthermore, because the cortex is exposed, one can topically apply on the cortex other pharmacological agents to gain insights on other aspects of regulation of the cerebral circulation. So for example, if we topically applied acetylcholine, we can look specifically for endothelium-dependent vasodilation. So using this model, then, we can look at all aspects of vascular regulation. So we use this model to look at the effect of hypertension in the brain in this mouse model. To elevate blood pressure, to induce hypertension, we used angiotensin administration. Angiotensin is very much involved in human hypertension. And so if you give angiotensin in very, very low doses that are not sufficient to increase blood pressure acutely, if you let the angiotensin go on for about a couple of weeks, you will see a gradual increase in blood pressure. And so this allowed us to monitor the changes in flow produced during this period of hypertension determination. And what we found was that the increasing blood flow produced by neural activity and by the endothelium were profoundly suppressed by the hypertension, even at 7 days after angiotensin, where there was no elevation in blood pressure still, highlighting the sensitivity of the cerebral circulation to the effect of angiotensin. Now, if you wait long enough, the mouse eventually is going to develop cognitive impairment. I know, I mean, the clinician among the audience, you know, will kind of laugh at this, you know, how we can measure cognition in a mouse. Well, I'm with you on that one, but there are ways to do it. And if you want to get a grant funded or a paper published, you better have it. Otherwise, you're not going to do that. But nevertheless, you know, you can see that there is evidence of cognitive impairment in the mice induced by this hypertension. Now, is this true in patients? So the answer is yes. So in this study, they compared visual stimulation increases blood flow in the visual cortex in patients with hypertension, which is this line over here, and with normotension, sorry, the top line is normotension, and patients with hypertension. As you can see, there is a suppression of the increase in blood flow produced by neural activity, also in humans, pretty much like what we saw in a mouse. Now, do humans develop cognitive impairment if they are hypertensive? Well, one of the most favorite studies that I have, that I cite on this topic, is the SEARCH study, which was done in Münster, Germany. They took about 2,000 people from the community. They were not patients, they were regular community dwellers. And they selected about 300-400 of them who had no evidence of neurological disease whatsoever. So they did a very detailed neuropsychological testing to assess cognition, and then they correlated a cognitive score, essentially how smart they were, with the systolic blood pressure. And as you can see, the higher the systolic blood pressure, the lower the cognitive abilities they were, even in the normal state, without brain lesions, without any other evidence of brain disease. So in our mouse model is pretty much predictive of what goes on in humans, both with respect to the vascular dysregulation and with respect to the alteration in cognition. Now, what mediates these effects of hypertension on the brain? Well, the problem is vascular oxidative stress. So production of free radicals in the cerebral, in the blood vessels of the brain. This is shown in this slide, in which we have CD31 is a marker of endothelial cells. And this is the top of the brain. And this is a penetrating arteriole diving into the substance of the brain, as it is here. If you give angiotensin, you have accumulation of this green reaction product, which is an indication of oxidative stress, which is centered on the blood vessels, but spread through the neuropil of the vessels. What is the source of these radicals? Well, the source is an enzyme called NADPH oxidase, which was first described in immune cells, neutrophils and macrophages. And this enzyme makes huge amounts of free radicals. So if you take a mouse which lacks one of the critical subunits of the enzymes, NOX2, you will see that angiotensin no longer is able to increase radicals and is no longer able to induce vascular dysfunction. So you essentially can fix, the mouse can remove the effect of hypertension by removing vascular oxidative stress caused by NADPH oxidase. Now, where do these radicals come from? Well, for many, many years, the field of vascular biology has focused on smooth muscle cells and endothelial cells as the major source of radicals. It turns out that this is not the case, at least in hypertension. In hypertension, the major source of the radicals is not the vessel itself, but are perivascular cells called perivascular macrophages, shown in green here, that are plastered on the surface of the blood vessel, and they are able to produce industrial amounts of free radicals, unlike vascular cells, which are not professional free radical producing cells. So what are perivascular macrophages? They are a class of brain-associated macrophages that come into the brain very early in development, pretty much when microglia comes into the brain, but it becomes attached to penetrating vessels and vein, artery and vein, and they pretty much reside in the perivascular space for years. They have a very, very slow turnover. If you look at them by electron microscopy, they look exactly like macrophages, except that they are plastered against the surface of the vessel. So they are endowed with NOX2, which is the free radical producing enzyme. They have angiotensin receptors, so raising the possibility that angiotensin may act on these macrophages and turn on free radical production, resulting in vascular oxidative stress. So we address this question. So to do that, we wanted to show first whether angiotensin gets in touch with the macrophage. Does it get there to engage the macrophage? To do that, angiotensin has to cross the blood-brain barrier. So we found that indeed angiotensin is associated with an increase in blood-brain barrier permeability. And you can see here biotinylated angiotensin, which shows in black at electron microscopy, kind of tracking along the tight junction and entering into the perivascular space. And then using CD206 as a marker of perivascular macrophages and biotinylated angiotensin, we could see that indeed there was an association between angiotensin and the perivascular macrophage. So it looks like angiotensin is able to engage the macrophage. But is this sufficient to induce the free radicals in the vascular dysfunction? To address this question, we used two different approaches. One was to get rid of the macrophages altogether in the brain. And how do we do that? Well, you can do that by administering a toxin called clodronate intraventricularly into the ventricles of the brain. Clodronate is prepared, encapsulated in liposomes, then is injected into the ventricles, is picked up by the macrophages, and then as a Trojan horse leads to a apoptosis of the macrophages. And these, you can see here, there is an 80% reduction of the perivascular of macrophages in the brain, shown by flow cytometry. No changes in the circulating white cells, and no changes in microglia. So this is a way to kind of selectively target the macrophage. And now we found, when we did that, we found that we could completely rescue the neurovascular dysfunction produced by angiotensin II. So it looks like the macrophage are required to have the vascular dysfunction, presumably through the production of free radicals. Now, in that experiment, we used a very kind of shotgun approach to just get rid of the macrophages altogether. But what we want to do is more selectively affect the source, the enzyme producing the radicals and the receptor, the angiotensin receptor that is activating the enzyme. Now, to do that, we had to use a different approach. We had to replace the macrophage that are normal in the brain with macrophages with altered genetic background. To do that, we can do a bone marrow transplant. Now, this is an example of this approach. In this particular case, we transplanted wild-type mice with a bone marrow containing GFP, which provide a green stain. As you can see, the macrophage, these perivascular cells are now all become green. And if you stain these for a macrophage marker, you will find them to be a bone marrow derived macrophages. So we have essentially, we can change the genetic makeup of these macrophages by using different kinds of bone marrows. So for example, we could transplant wild-type marrow into wild-type mouse as a control. Or we can transplant angiotensin receptor deficient marrow or NOX2 deficient marrow into wild-type mice, in which case we would change the makeup, the genetic makeup of the macrophages by eliminating either the AT1 receptors or NOX2. So if we do a wild-type to wild-type transfer, we give angiotensin, we have neurovascular dysfunction as anticipated, all right? But if we transplant an AT1 receptor negative marrow or a NOX2 negative marrow, we no longer have neurovascular dysfunction. And importantly, we also rescue the cognitive dysfunction in a mouse. So it looks like the macrophage AT1 receptors and NOX2 are in the macrophages are critical for these effects. So the data suggests that in angiotensin hypertension, angiotensin engages the perivascular macrophage, through the angiotensin receptor leads to activation of NOX2 and to production of massive amounts of radicals that alter all aspects of the regulation of the cerebral circulation. Now let's move on to Alzheimer's disease. We know, it's been known for a while that Alzheimer's patients have alteration in blood flow, have alteration in cerebral blood vessels. And also there's been known that Alzheimer's disease and vascular brain diseases have similar risk factors. Hypertension, dyslipidemia, obesity are all risk factors for Alzheimer's as they are for cerebrovascular diseases, suggesting mechanistic similarity. Furthermore, the NAN study showed that the small ischemic lesions located in the basal ganglia, which would have no impact on cognition, when these very little lesions were associated with a mild degree of Alzheimer's pathology, it amplified tremendously the impact on dementia. So some have suggested that there is an interaction, this is a synergistic interaction between the ischemic lesions and the Alzheimer's pathology that kind of synergizes and aggravates the expression of the dementia tremendously. So these studies have raised the possibility then that the beta peptide and perhaps even tau, in addition to having the well-established effects on neurons producing neural dysfunction and synaptic dysfunction, which is very well established, they may also alter vascular dysfunction, providing another kind of negative impact on neurons by starving them of the needed delivery of oxygen, glucose, and clearance, and so on. So a number of years ago, I had the opportunity to test this hypothesis that A-beta may have also vascular effects using a model which was just recently developed at the time, which is a mouse model of overexpression of amyloid beta. These are called TG2576 mice made by Karen Shaw at the University of Minnesota, where I was at the time. And so we were able to study these mice when they were very young adults, and to look at whether when they were normal cognition, they had no pathology in the brain yet. They had elevated A-beta, but no pathology yet. So we did the neurovascular assessment in these mice, and what we found was that there was a profound alteration of all the mechanisms regulating the cerebral circulation. Neurovascular coupling, endothelium-dependent relaxation, and there was essentially no autoregulation in these mice. So there was a profound effect at the time where the mice were not yet sick. So this suggested that perhaps the vascular alteration was an early aspect of A-beta pathology before there was a accumulation of plaques and other tangles and so on, and before there was a decline in cognition. So this remained an hypothesis for a number of years until a few years ago, from the ADNI study, which is one of the largest collection of imaging studies in Alzheimer's patients, it was found that the earliest biomarker for Alzheimer's disease is the vascular alteration. So in the Alzheimer's mouse, as in patients with Alzheimer's disease, vascular dysfunction is one of the earliest manifestations of the disease, which obviously may have a biomarker value. So the point is that what we saw in the mouse is also relevant in humans. Now, what causes alterations of vascular dysfunction in Alzheimer's? Well, it looks like it's also oxidative stress. And you can see the brown reaction product indicating oxidative stress in the PL arterioles in this Alzheimer's mice. Now, the enzyme making the radicals is NOX2, pretty much like hypertension, all right? However, the mechanism of synthesis of activation of NOX2 are different because we have an innate immunity receptors in the case of A-beta that binds A-beta and activates this enzyme and also activates inflammation, leading to both the inflammation, oxidative stress, and neurovascular dysfunction. To make a long story short, in hypertension, as we found in hypertension, in Alzheimer's models as well, A-beta activates these receptors on these macrophages surrounding the blood vessels, leading to activation of NOX2 and free radical production, which again produces a massive dysregulation of all aspects of the cerebral circulation. So what I showed you so far is that they are in hypertension and in amyloid pathology, and also in ApoE4 expression, which I'm not going to discuss today. The perivascular macrophage is the critical cell that makes the free radicals that are needed to alter all aspects of vascular regulation: endothelial regulation, the increase in blood flow produced by neural activity, autoregulation, and blood-brain barrier disruption. So this is a massive dysfunction of all aspects of the regulation of cerebral circulation. Now we're going to switch gears, and I'm going to show you another source of cognitive impairment in which things are quite different. And this has to do with excess dietary salt. It's been known for many, many years that dietary salt is a cause of stroke and dementia. And here we have the number of stroke deaths for urinary sodium excretion, which is an index of dietary sodium, probably the most accurate index of how much sodium you eat through the food in the different nations in the world. As you can see, the higher the average consumption of salt, the higher is the risk for stroke death. So this is just one of many, many studies. And all of these studies obviously led to the public health system to say, okay, we got to cut down on salt. Okay, so there was a massive campaign encouraging people to cut down salt. However, this campaign led to backlash from both the kind of the scientific literature as well as from the lay people, because they said that, you know, it's not really absolutely certain that salt is doing all these things. And there was one study showing that if you restrict salt too aggressively, you can increase mortality. So the public was very confused. So when that happens, you know, usually the matter is referred to the Institute of Medicine of the National Academy. And so there was a report from the Institute of Medicine that, as often happens, said, okay, guys, we don't really know what's going on, so you might as well do some more work on this. So that was the advice from the Institute of Medicine. And so a number of years ago, we decided to investigate what happens to the brain when you feed too much salt to an animal. So we took mice again. We fed them very high salt levels of dietary salt, and then we looked at all kinds of brain blood flow related parameters. Now, importantly, when we gave all this salt to the mice, HST stands for high salt diet, there was no change in arterial pressure. And we knew that because this particular kind of mouse we used does not have salt-dependent hypertension. Like 30% of humans, they do not have, they don't develop hypertension when you give them salt. However, they had about a 30% reduction in blood flow to the brain, as shown here. And they had an attenuation, decrease in blood flow produced by the endothelium, so endothelial dysfunction. However, to our surprise, there was no attenuation of the increase in blood flow produced by neural activity. And this was very surprising to us because usually if the vessel falls apart, everything doesn't work. Here instead, we have a selective problem with the increase in blood flow produced by endothelium. And so I was skeptical whether this would have an impact on the brain. But as often happens, I was wrong. Because feeding the high salt diet for a couple of 3 months or so produced cognitive impairment in the mouse. So how do we know that? We know because the mouse is no longer able to recognize novel objects. The mice, the rodents love to see new things, and they don't pay attention to stuff they've seen before. So these mice, couldn't tell whether something was new or old. And most importantly, the mouse could not build a nest. Now, nest building is an activity of daily living for the mouse. And so if the mouse cannot build a nest, it's really very much messed up. And so that's what happened in these mice. So despite this kind of relatively minor changes in flow regulation, we had this massive cognitive dysfunction. So the fact that acetylcholine response was reduced, as I told you at the beginning of the talk, suggests that perhaps nitric oxide is not being made in endothelial cells. So we took the blood vessels of the brain, the microvessels, in animals from a normal diet and animals receiving high salt diet. We put them in a dish and we measured their ability to make nitric oxide. And what we found was that the resting level of nitric oxide in these vessels were in high salt diet were reduced, okay? And also acetylcholine was unable to increase nitric oxide production in response to eNOS activation. So there was something wrong with the ability of the endothelium to make nitric oxide. As I told you, the endothelium make nitric oxide from an enzyme called endothelial nitric oxide synthase, which is regulated by calcium, but also by phosphorylation that sets the gain of the enzyme. So there is an excitatory phosphorylation that increases the gain, serine 117, and an inhibitory phosphorylation, threonine 495. What we found was that the high salt jacked up the inhibitory phosphorylation so that eNOS was not efficiently making nitric oxide. And so, in fact, if we rescued nitric oxide production by providing L-arginine, which is a precursor for nitric oxide, we could completely rescue the deficit in nitric oxide production in the vessels, and we also could make the mouse normal cognitively, all right? We can rescue cognition and NO production if we provide the nitric oxide that is missing. Now, this was very nice and very well understood. However, the problem was that how do you get from high dietary salt, all right, to have endothelial dysfunction in the brain? So how does that happen? So we were struggling with this for a couple of years until two papers were published in Nature in which they showed that high salt diet has a tremendous impact on immune cells in the gut. It leads to the expansion of a group of lymphocytes called Th17 cells that are from the gut are able to kind of spill over to other organs. So we thought that maybe what happens here is that this Th17 mechanism could also be effective in the high salt diet with respect to cognitive impairment. So we went on, and here I'm summarizing, several years of work, and looked at whether this was true also in our model. And indeed, high salt lead to an expansion of IL-17 cells using an IL-17 reporter mouse in the gut, as shown here, and also by flow cytometry. This expansion of IL-17 was so massive that circulating level of IL-17 were increased. So there was more IL-17, this is a cytokine that is vasotoxic, circulating into the blood vessels. And what we found was that IL-17 was able to activate an enzyme called Rho kinase in endothelial cells and induce this phosphorylation of eNOS that blocked the nitric oxide production. And so, in fact, if we administer the Rho kinase inhibitor to the mice to block this enzyme and to block the effect of IL-17, we could completely rescue the blood flow change as well as the cognitive dysfunction in the mice. So the data suggested that in this particular case, we have this gut-brain axis in which there is expansion of Th17 cells, increase of IL-17, activation of Rho kinase, inhibitory phosphorylation of endothelial nitric oxide synthase, and deficit of nitric oxide. Now, more recently, we became interested in that how does the NO deficit lead to the cognitive impairment? Okay, is it through the blood flow or is it through something else? Now, we were not impressed with the changes in blood flow that we saw, that kind of intermediate in neurovascular dysfunction. So we were thinking maybe there is something else going on. So a few years ago, there was a paper published in Cited Researchers that indicated that nitric oxide, specifically from the endothelium, was able to keep phosphorylated tau, inhibited. Now, as you know, tau, particularly phospho-tau, accumulates in the brain of patients with Alzheimer's disease and is a very major source of cognitive impairment because phosphorylated tau tends to aggregate and to eventually form neurovascular tangles, and which are thought to be part of the problem. So NO had this ability to inhibit this production of tau, particularly by reducing CDK5 activity. So we thought perhaps what's going to happen here is that NO may inhibit calpain, which is the enzyme that activates the tau formation, by nitrosylating it, by adding an NO group, so that when there is a reduced production of nitric oxide from the endothelium, you have a denitrosylation of calpain and activation of CDK5 activity, leading to phosphorylated tau, which then leads to misfolding, aggregation, and eventual neuronal dysfunction. So it could be that this tau may be what's causing the dysfunction in this high-salt diet model. So we went on and to test this hypothesis. And so what we did, we measured phosphorylated tau in the brain of the animals treated with high-salt diet, both in hippocampus and their cortex. And what we found was that there was indeed an increase in phosphorylation of tau in many of the epitopes, which are well known to be associated with cognitive dysfunction. We also saw accumulation of phospho-tau in the entorhinal cortex and hippocampus. And importantly, fractionation studies showed that the tau that was accumulating was not free tau, but was partially aggregated, although tangles were not observed. So there were tau multimers that were aggregating into the cortex and hippocampus. And as predicted, we found that there was high salt, reduced calpain nitrosylation, activated calpain, and also activated CDK5, which is the enzyme that is responsible for this tau phosphorylation. Now, the question is now, is this tau phosphorylation biochemistry responsible for the cognitive impairment? Keep in mind that, phosphorylated tau is not always bad for you. So there are instances in which, there is accumulation of phosphorylated tau and the brain is protected by it. So having tau in the brain doesn't mean that this going to cause trouble. So to really show that this was causing the problem, we used antibodies against tau, pretty much like the same antibodies used in clinical trials for Alzheimer's disease. And what we found was that if we gave these antibodies to the mice on high-salt diet, we could completely rescue the cognitive dysfunction that we had produced by the high salt. So if we suppress tau, we can improve cognition. So the next question is, what happens to blood flow? Is blood flow also rescued, or blood flow is not rescued? And so that's what we did. We measured resting blood flow and the increase in blood flow produced by acetylcholine, the endothelial dysfunction. And what we found, to our surprise, is that despite the fact the mouse was cognitively normal, flow was still reduced as much as it was before. And the endothelial cells were dysfunctional as much as they were before. So the data then suggests that in this model, the NO deficits causes a flow reduction, but the flow reduction is not responsible for the cognitive impairment. Rather, the NO deficits activates calpain by denitrosylating calpain, leads to CDK5 activation, accumulation of phosphorylation tau, and cognitive impairment. And in fact, if we block this tau effect, either with antibodies or in the tau knockout mouse, which I did not show, you have still the flow reduction caused by the NO deficit, but you have normal cognition because the tau is what really causing the cognitive dysfunction. So this data then provides an evidence that really in this particular case, flow doesn't seem to have anything to do with the cognitive impairment, but is rather the tau produced by the deficit of NO that is playing a role. Now, you may be wondering whether there is any human evidence that there is some link between salt and cognition, all right? Well, obviously, people have not been looking at the connection between salt and cognition as much as they've been looking at cardiovascular effects. But there is some evidence that this may be indeed the case. So in this case, we have cognitive function, mini-mental state, versus dietary salt. So the higher the salt consumption, the lower the mini-mental state. Okay, suggesting maybe some kind of link. Now, there is a wide variety of salt consumption among world population, right? Is there evidence that the nations in which there is more salt consumption, for example, in Central Asia, they also have a higher incidence of cognitive impairment? Again, there isn't much literature, but there is this evidence published last year in which in Kazakhstan, which is the region of the world where the highest salt consumption is observed, there is an increase in prevalence of mild cognitive impairments. So maybe there could be a signal there suggesting a link between high salt and cognitive impairment. Now, what we really want to know is that if you reduce salt intake, are you going to improve cognition, all right? Again, the evidence, there is not much published on this subject, but one recent paper in which they tried a DASH diet, which is a diet that is meant to reduce hypertension and also reduces, is low in sodium. They attributed the improvement that they saw in these patients to the reduced sodium in the diet. So the other factors in the DASH diet did not have a role, except for the reduction in sodium that the patients were taking. So the data suggests that even in humans, there may be some link between the dietary salt consumption and cognitive impairment. Now, to wrap it up now, I'm going to summarize what I told you by saying that there are conditions in which a neurovascular dysfunction leads to cognitive impairment, in which, there's a massive disruption of all the factors regulating the cerebral circulation, produced by production of free radicals from perivascular cells called perivascular macrophages. And this seems to be the case in amyloid pathology and in the pathology associated with hypertension. However, there are other conditions, like in a dietary salt, where there is a gut-brain axis in which the immunological effects produced by the high salt leads to this IL-17 mediated suppression of nitric oxide made from the endothelial cells, which does not cause dementia by reducing flow, like in this case, but by producing tau phosphorylation, which is truly responsible for the cognitive impairment. So what is the take-home message from this mechanistic study? Is that there is neurovascular dysfunction does not cause cognitive impairment through the same mechanism as we always believe. It's not always blood flow that is the problem. There are also other aspects of vascular function which have to do with the homeostatic function of the blood vessels beyond the delivery of oxygen and glucose that play a role. And the fact that endothelial nitric oxide is required to keep tau out of the way is very, very important and may provide insights to new approaches in which we can counteract the deleterious effects of vascular dysfunction of cognition. And in closing, I'd like to thank my colleagues who did all the work, Josef Anrader, Karel O'Kreiner, and Lebach Park, and Dr. Farag who did all the work on tau and hypertension, and also the funding agency for providing the resources necessary for this. And thank you for your attention.
Thank you so much, Dr. Iadecola. I think this was a phenomenal tour de force, and we received a few questions. We're not gonna let you get off lightly. So the first question I wanna pass to you has to do with the issue of happy medium. Have you looked at low salt as being having problems, or how low can you go, in other words, as they say in Italy?
Yeah, well, if you go too low, you're gonna be in trouble. All right.
All right.
So salt is one I think is the example, is a perfect example of precision medicine, right? So depending on what age you have, depending on what kind of body constitution you have, depending on whether you're an athlete or a sedentary or whether you're young or old or whatever, it's going to have a profound effect on your salt intake. So that's why a number, is not going to fit all. So an athlete on the American Association recommended level is going to be in trouble. Because that's not enough salt, because he sweats a lot and he loses more salt and so on. So we need to have a dedicated, one-by-one approach to assess the salt level. However, in as much as we know that excessive salt intake comes from processed food, it's not really the food that you add to your, at home. It's when you eat to the restaurant, when you do the soup in a can, and that's what really causes the salt. 70% of the excess salt comes from processed food. And so that's where really we need to pay attention. It's not what you put on your meal at home, all right? It's really what the industry is feeding you without you knowing that needs to be addressed.
So a follow-up to that question, Cos, is what is the best biomarker of optimal salt intake? Is it the blood pressure?
There is one biomarker in 30% of the people. So if you give a salt load to 100 people, and only 30 of them will develop an increase in blood pressure. That's called salt-sensitive hypertension. Now, if you are hypertensive, however, 50% of you will be salt sensitive. That's why the first thing that the doctor does, when you with high blood pressure, is exercise and reduce salt. All right, because those are things you can do and lower your blood pressure if you happen to be salt sensitive. But the measuring salt consumption is very, very tricky because it changes during the day, oscillates during the day. So spot checking urinary sodium is not going to be the preferred way to do it. And the best studies, they do 24-hour urine collection, which, as you know, is very, it's not easy to do accurately. And that's really the way some of the, the best epidemiological studies are doing it.
So one other question, you probably have studied 10,000 mice if we kind of figure out what you're doing there. There are female and male mice. So do you have any signal about a different effect in the sexes?
Yeah.
Between the effect of salt, the effect of blood pressure on dementia?
Very, very important question. So there is a profound sex difference in hypertension. As you know, women are protected from all cardiovascular risk factors until they hit the menopause. When they hit the menopause, they have even more trouble than men do. So that's a very important concept, particularly with hypertension. So for example, if you give angiotensin to a young female mouse, will not get hypertensive. Pretty much like a woman, during the reproductive stage of life, will be protected from cardiovascular risk factors. In salt, however, we saw an equal effect between males and females. In amyloid has an equal effect in males and females. There is also an important sex difference in ApoE, in the effect of ApoE on the brain. So I think this emphasis that now we have on sex difference is very, very important. With respect to vascular risk factors, you can assume that studying a young female is almost worthless because, it's not really something that reflects the disease incidence. And so studies should really look at all females, and then you're going to have a more representative example. Yeah.
We have a very special question from last year's Safadi lecturer, who is a very good friend of all of us, Dr. Walter Koraches is actually attending the talk as we speak from the NIH. Sorry, Walter, we cannot see you, but we can get your question. And I know Mr. Safadi probably wants to say hi as well. The question that Walter is asking is, what do you know about the pharmacodynamic relationship between high salt into the gut and tau? So the tau phosphorylation, meaning are these cognitive changes that result from this reversible or not? If so, how long does the negative effect last?
Okay, that's Walter. I am honored to follow you. If I knew that I were following you, I would have refused to give this talk. All right, I cannot give an NINDS level talk like the one you do. But thank you. The NINDS is funding all our SALT work, and they've been very obviously supportive of these efforts. So to your very important question, so the effects of salt on the brain are dose dependent. So the more you give, the more you get on tau, and the tau goes away within a month after stopping the salt administration. After 3 months of salt, if you stop it, the mouse a month later will become normal, which means there is hope for the rest of us, all right? And so it supports also the result of that study that I showed you, the DASH-DAT study, in which, curbing salt administration was able to have some positive effects. However, there is an interesting biological phenomenon in hibernating animals, all right? So hibernating animals, when they go to sleep, they phosphorylate their tau. And let's say an hibernating bear does that, many hundreds of, maybe, 50 times or so, 60 times throughout his life, his rare life. It turns out that the more you turn tau on and off, the more it tends to accumulate. So the hibernating bear, when he gets very old, is going to have tangles that did not have when he was young. So I think turning the system on and off, or sustained upregulation, may have long-term effects which may no longer be reversible. The caveat of this is that, as Walter knows well, the tangles are not really what's causing the problem, because you can improve cognition without improving the number of tangles. And it's probably a lower degree aggregate of tau that is producing the problem. And so with that caveat, however, probably on and off or prolonged sustained may lead to more permanent effects. Very important question.
Several questions actually can be summarized in one. Having to do with, we talked a lot about the negative effects, whether of NO production, of some of the enzymes, etc. But these enzymes and that process of production, evolved through evolution or whatever to do something that is good. So is it just about inhibiting all of these things? I mean, what do they really do for your good? The same even applies to Rho kinase. Everything we hear is Rho kinase is bad. So why is it there?
Absolutely. Very, very important question. So first of all, the salt story is a very interesting one, because the cave, as you know, salt in nature is very, very poor. There's very little salt around, right? So the caveman used to assume like 10,000 times less salt than we do now, all right? So we have kind of evolved into this high salt diet, which was not there, when there was not much salt around. And it mainly started first with the invention of salt being able to preserve food, and then people start to eat much more salt. And more recently, the introduction of processed food. So both of those events have made so that's an abnormal situation in which the humans were not meant to assume that much salt. Now, with respect to the Rho kinase and IL-17 and so on, so those are obviously regulatory mechanisms. They are part of the immune system, and immune system is very important to maintain homeostasis. However, salt induces dysregulation of the immune system, so it's almost like an autoimmune response, triggered, through some mechanism which we still have to clarify, but clearly is not really an exaggeration of a normal response, but essentially is a pathological response coming from alteration of the immune system. And if you block Rho kinase, all right, the blood flow goes very high in the brain, and you may have hemorrhages, all right? So you need the right level of Rho kinase, but if you overactivate it, all right, you're not gonna be fine. Okay, an MDA receptor is the same. Okay, you can have an MDA receptor, you can have, ischemic injury if you put too much an MDA receptor activation, but you need the right level of an MDA to remember and to think and so on. So it's all in the homeostatic balance that all of this effect, these pathogenic effects, disrupt, therefore evoking a pathogenic aspect out of this.
So it goes on to say that we need really better biomarkers of the happy medium again. There is, I want to end with really two more questions, and then give the chance to Mr. Safadi if he has any questions in particular or concluding comments. I'm sorry we are not able to really answer all the questions that have come up, but there is one question that I think is very cogent to the era in which we are living. There was a recent eLife paper linking ACE and COVID to vascular dysfunction through changes in bradykinin levels. And we are wondering if this might contribute to brain dysfunction following COVID infections.
Linking, can you repeat linking what?
Yeah, so it's actually the paper linked ACE and COVID to vascular dysfunction through changes in bradykinin levels.
ACE, yeah.
Yeah, ACE.
Okay, so ACE is one of the receptors for angiotensin. Right. ACE2 actually is it turns out that COVID, the virus, uses ACE2. Well, COVID-like viruses use ACE2 to enter the cell, to infect the cell, all right? Now, it's not the only receptor. There is also other receptors. Now, one idea is that, when the virus binds to ACE2, it gets internalized. So leaving angiotensin to activate other receptors like the AT1 receptor that is bad for you. So that's a possibility that there is this interaction between angiotensin. In fact, angiotensin levels, there is one report of circulating angiotensin level being increased in patients with COVID-19, because you removed one of the receptors that would have otherwise captured it. I have to make a comment about the effect of COVID-19 on the brain, and I've reviewed the literature recently, and there is no real direct evidence that there is a neurotropism of the virus for the brain, like there is for herpes, like there is for polio or whatever. However, it seems like the immune dysregulation that this virus induces, mainly because of the effects on the lungs, are really the one that lead to the cytokine production, and then may act on the brain to produce a cytokine syndrome, cognitive impairment and so on, that may be reflected in some of the manifestation of neurological manifestation of COVID, most of which can be explained by the context. The patients are very, very sick. They are hypoxic. They have some of them have renal failure. They have a profound, hypercoagulable state. That all of those, obviously put to that, many of them are intubated, and 80% of people intubated, they won't have delirium. So, it's been a little bit hyped up, But certainly this impact of ACE2 on the endothelium, which, by the way, the human endothelium doesn't have. Many human studies, single cell transcriptomic studies have not shown. However, the endothelium does have other receptors like neuropilin, which also may be able to bring the virus inside the endothelium. So the question, it's too early to say what's going on.
So we're not going to worry that if you didn't have these very extreme illnesses, probably there is not a direct effect of COVID on the.
That's what right now, the evidence is not, most of the studies are not reviewed yet. As you know, they're all in the bioarchives, and that's what we can surmise right now.
So final a couple of questions. So given that there is a big link between midlife hypertension and dementia, is there a point of no return in animal models? Meaning a point where this dementia has set in and is not reversible? Yeah.
So in animal models, you can bring it. You cannot bring it forever because the mouse cannot sustain. You cannot give it. But there is a model called a BPH mouse where there is a spontaneous hypertension. Until they're about a couple of months old, you can reverse some of the effects. But then beyond that, you cannot do them anymore. So in humans, probably is a similar situation in which there is this early hypertension that for some reason, has a cumulative effect that only 20 years later can be evidence to cause, cognitive impairment, often in association with tau and A beta, as some of the studies have shown that, hypertension may favor the accumulation of tau and A beta in the brain. So there will be this kind of interaction between neurodegenerative and cardiovascular factors leading to the dementia.
Truly final question, then I will ask Mr. Safadi to comment. So are there any salt-independent effects of blood flow and blood pressure on dementia? And particularly, there is this syndrome of post-cardiac bypass, cognitive impairment. Is this a flow-related issue, or is this bad stuff happens during clamping and unclamping?
Yeah, obviously, yeah. There is a microvascular, there is a micro-occlusion, and there is reperfusion injury, and there is all kinds of stuff like that. One idea that is kind of being surfacing is that during catastrophe, severe systemic disease, the immune system gets tickled in the wrong way, and there is a lingering immune dysfunction that may traffic to the brain and may lead to a kind of chronic, non-resolving inflammatory process that in some conditions, like for example in stroke, they may be involved in subsequent cognitive impairment. So the flow may be triggering the things at the beginning, but then there is something else that takes over that may sustain this, and the immune system is perfect for that because it's got a memory, all right? And so it's perfect to kind of perpetuate these damages forever, yeah.
So I really want to take this opportunity to thank you for having defined really a number of steps in this very complex equation, that many of which are going to be amenable to measuring them through biomarkers and altering them through specific therapies, and gives us a little bit better of a scientific handle on what has been a big myth for almost 100 years in our society about blood vessels and dementia. Mr. Safadi, do you have any final comments or questions or thoughts that you would like to share with us before we conclude this wonderful, wonderful forum? I would want you to go ahead and yeah, you are good.
Well, I'd like to thank Dr. Iadecola for everything he said. I mean, I wish I could understand it all, but I'm sure I'm gonna go home and change my diet. This is for sure. I'm gonna cut my salt intake because I have hypertension. So thank you very much. At least I got that from you. But all the other final details, I look forward that I meet you. You know, when I come to the States, and maybe we can talk more about it.
Absolutely, it would be my honor. Would be my honor.
I really appreciate it. I really appreciate it. Thank you so much.
Thank you.
Thank you, everybody, for joining us. I want to thank the Dean for hosting us, and Cos, you are truly amazing. We are very, very proud and grateful for your presentation. And Your Excellency, thank you for gracing us with your presence. Thank you. This concludes the forum.